There is a worm at the bottom of the sea, and it has no mouth, it has no stomach, it has no gut, and no way to pass anything through a gut, even if it had one. It grows to the length of a grown person lying down, nearly 2 meters, sometimes a little more. And it does this in under 2 years, which makes it one of the fastest growing creatures of its kind anywhere in the ocean. And yet it cannot eat, not in any way you would recognize as eating. It has no opening to take food in and no opening to let anything out. By every rule we were ever taught about how an animal is supposed to work, this worm should not be alive. And it is not just alive. Down there in the cold and the dark, 2.5 kilometers below the waves, where the weight of the water overhead would crush you flat, this mouthless worm thrives. It grows in great swaying forests of pale tubes, each one tipped with a plume the color of fresh blood. And it does all of this without ever once feeding the way you or I, or almost every animal you have ever met must feed to stay alive. Welcome to Aurelius Explainer. Settle in. There is nowhere you need to be tonight, and nothing you need to solve. Let your shoulders drop a little from wherever they have been holding all day. You can let your eyes close whenever they want to. You do not have to follow every word because there is no test at the end of this, and no part of the story will be lost if you drift off somewhere in the middle of it. The whole point of tonight is the drifting. We are going to go slowly down into one of the strangest places on this planet, a place most people never think about even once in their whole lives, and we are going to take our time getting there. The descent is the whole journey, so breathe out and let me carry the thinking for a while. The worm is real. It has a name, a proper Latin name, the way all catalogued creatures do, and its name is Riftia pachyptila. People who know it simply call it the giant tube worm. It lives clustered around hot cracks in the floor of the deep ocean, fastened to bare volcanic rock, in places where the seawater itself comes pouring out of the seabed, warm and strange and full of chemicals that would kill almost anything you have ever known. And the question that hangs over it, the question that has for it since the first stunned scientist laid eyes on it, is so simple a child could ask it. If it has no mouth, then how on earth does it eat? We will get to the answer. The answer is one of the most beautiful pieces of biology ever uncovered, and it took years for anyone to believe it. But to understand why the answer matters so much, you have to first understand how we found this worm at all, and just how completely it overturned what everyone thought they knew. Let me take you back to a particular dive. It was February in the year 1977. There is a small, deep-diving submersible called Alvin, a research submarine built to carry a tiny crew down to depths where ordinary vessels would be flattened like a soda can underfoot. Its full designation is DSV Alvin, the letters standing for Deep Submergence Vehicle, which is exactly what it is, a vehicle made for going deep. On this particular expedition, Alvin was diving on a feature of the seafloor called the Galápagos Rift, a long seam in the ocean bottom out near the Galápagos Islands, the same islands that once gave a young Charles Darwin so much to think about. The submarine was descending to around 2,500 meters down. That is 2.5 kilometers of black water stacked overhead. At that depth, there is no sunlight at all. None. The last faint blue traces of the sun give out far above, perhaps a few hundred meters down, and below that there is only a darkness so total and so old that most of the creatures living in it have never known anything else. Now here is a detail I want you to hold on to because it tells you everything about what was about to happen. The people who planned this dive were not biologists, they were geologists. They were scientists who studied rock and heat and the slow machinery of the planet itself. They had gone down to the Galápagos Rift expecting to find warm water seeping out of the seabed because the physics of the spreading ocean floor said warm water ought to be there. They wanted to measure it to study the chemistry of the rock and the heat of the planet's interior leaking out through the cracks. They were looking for geology. There was no biologist aboard that first descent. Nobody on that dive had come looking for life, because nobody, not one single person, expected to find very much of it down there at all. And then the lights of the submersibles swept across the seafloor, and the scientists pressed their faces to the little portholes, and what they saw made no sense. There was life everywhere. Not the thin, scattered, half-starved life you might expect on the floor of the deep sea, where most of the bottom is a kind of cold desert, dusted with whatever scraps drift down from the sunlit world far above. This was an explosion of living things. Great clusters of white tubes taller than a person, swaying gently, each one tipped with that vivid red plume. Clams the size of dinner plates wedged into the cracks. Crabs picking their way across the rock. Mussels in heaps, a riot of living, breathing animals packed densely together in the warmth pouring out of the seabed in a place that should, by every rule, have been almost empty. The geologists, by some accounts, did not even have the right equipment aboard to properly preserve the creatures they were hauling up. They improvised. They are said to have used vodka from the ship's stores to keep some specimens, because no one had imagined needing the proper biological gear for a geology cruise. They had come for rocks and stumbled into a garden, and a garden is exactly the right word, because it raised an impossible problem. Every garden you have ever seen, every forest, every meadow, every field of waving grass is ultimately fed by the sun. This was one of the bedrock truths of biology, as solid and unquestioned as anything in the whole of science. The sun shines, and plants and algae and certain bacteria capture that light and use its energy to build sugars out of air and water. That process is called photosynthesis, and it is the foundation under almost everything alive. The grass uses the sun, the deer eats the grass, the wolf eats the deer. Follow any food chain on Earth back far enough, all the way down to its root, and you will arrive at sunlight. Even in the deep sea, the assumption went, the animals down there were living on leftovers, on the slow rain of dead matter sinking from the sunlit surface, a thin and meager fall of crumbs from a table set far above by the sun. No sunlight, no food, no life. That was the rule. It was the whole structure of how the living world was understood to work. And here, 2.5 kilometers down, in water that had not seen a single ray of sunlight for the entire history of the place, was a thriving, crowded, abundant community of large animals, packed more densely than seemed possible anywhere, let alone in the lightless deep. The crumbs falling from above could not possibly have been enough. There was simply too much life, too much flesh, too much hungry biomass, crammed into that small, warm patch of seabed for it to be running on scraps from the sun. Something else was feeding all of this. Something the rule did not account for. And whatever it was, it meant that the most basic assumption in biology, the idea that the sun feeds everything, was not the whole truth. There was another way to make a living on this planet, a way no one had ever properly seen. That other way has a name, and it is the quiet hinge on which this entire story turns. It is called chemosynthesis. Hold the word loosely if you like. It does not matter if it slips away from you. What matters is the idea underneath it, and the idea is simple and strange and wonderful. Photosynthesis, the familiar one, builds life out of the energy of light. Chemosynthesis builds life out of the energy of chemistry. Instead of catching sunlight, certain microbes catch energy from the raw chemicals dissolved in the water itself. They take simple, lifeless, inorganic substances pouring out of the cracks in the seabed, and they pull energy out of them by rearranging them, and they use that energy to build the stuff of life, the carbon-based molecules that make up every living body. Where a blade of grass uses photons, these microbes use the chemistry of the rock and the vent. They are the green plants of the deep, except that there is nothing green about them, and there is no light for green to matter. They sit at the very bottom of this whole crowded food web, the primary producers, the makers, the ones who turn dead chemistry into living tissue, and everything else down there. The worms, the clams, the crabs, the mussels, every animal in that impossible garden depends on them, directly or at one remove. They are the grass of a world without a sun. And now we come to the part that I find the most haunting, the detail that turns this from a curiosity into something almost unbelievable. What is the chemical these microbes are feeding on? What is the fuel at the bottom of this entire ecosystem? The substance these tiny creatures break apart to harvest the energy that keeps a 2-meter worm alive. It is a chemical called hydrogen sulfide. You may not know the name, but you know the smell. Hydrogen sulfide is the gas that smells of rotten eggs. It is the stench of a sulfur spring, the reek that rises off stagnant marsh mud when you disturb it. And it is poison. To you, to me, to nearly every animal that walks or swims or crawls on this planet, hydrogen sulfide is a toxin. In high enough concentrations, it will kill a person quickly. It attacks the very machinery your cells use to breathe. It is one of the substances your body has spent millions of years learning to flee from, because the smell of rotten eggs is, deep in your instincts, the smell of something that can stop your breath. And that poison, that rotten egg gas that your body recoils from, is the bread of life at the bottom of the sea. The whole flourishing community down there, the swaying forests of tube worms and the heaped beds of clams, it is all built from the ground up on a foundation of poison. The microbes drink in the hydrogen sulfide rising from the cracks in the seabed, the same molecule that would suffocate you, and they pull it apart, and they live on it, and from that they make the food that feeds everything else. Life there did not just find a way to survive without the sun. It found a way to survive by feasting on something that to the rest of us is death. Sit with that for a moment, gently, as you lie there in the dark. The thing that poisons us is, somewhere far beneath the waves, the thing that gives life. The rule was never as simple as we thought. There is more than one way to be alive on this world, and one of them runs on the very stuff we run from. Before we go any deeper, and we are going to go a great deal deeper before this night is through, let me ask you something the way I like to ask, gently, with no urgency to it. I am always curious where in the world you are listening from tonight. Some of you are far north where the dark comes early and stays late. Some of you are somewhere warm with a window open. Some of you are in a city that never quite goes silent, and some of you are out where there is nothing but the sound of the dark itself. If you feel like it, and only if it costs you no effort at all, you could leave a small note of where you are, just so the sea of this strange little story feels a touch less lonely. And if you find that this kind of slow journey into the deep helps carry you towards sleep, you might quietly subscribe, so the next one finds its way to you when you need it. There is no rush in any of it. You can do it tomorrow or never. For now, just keep drifting down with me, because we have left a question hanging, and the answer to it is one of the loveliest things I know. The question is the worm, the mouthless worm, Riftia, the giant tube worm with no mouth and no gut, and no way to take in food, growing 2 meters long in a place where the only thing to eat is a poison that only microbes can stomach. If the microbes are the grass of this world and the worm cannot eat, then how does the worm get fed? How does a creature with no opening into its own body live, and not just live, but flourish in the richest part of this dark garden? Here is the answer, and it is the kind of answer that makes you put down whatever you were doing the first time you truly understand it. The worm does not eat the microbes. The worm becomes their home. Inside the body of Riftia, deep within that long, pale tube of flesh, there is a special organ. It has a name, the trophosome, and the name itself is a small poem because it comes from old words that mean, roughly, the feeding body, the nourishing body. The trophosome is not a stomach. It does not digest food that has been swallowed because nothing is ever swallowed. Instead, the trophosome is a living farm. It is packed, absolutely crammed, with bacteria. Not bacteria that have wandered in by accident. These are the very chemosynthetic microbes we were just speaking of, the ones that live on hydrogen sulfide, the grass of the deep. The worm carries them inside its own body, in numbers that are almost impossible to picture. In a single gram of trophosome tissue, a piece of this inner organ, smaller than a sugar cube, that can be on the order of 1 billion bacterial cells. 1,000 million tiny living things packed into a fragment of flesh you could balance on a fingertip. The whole organ is a teeming, dense, internal pasture of microscopic life. So the worm has solved the problem of having no mouth by simply not needing one. It does not hunt for food in the water around it. It does not chase or graze or scavenge. Instead, it grows the food inside itself. It is a farmer that has become its own farm. It takes in, through that beautiful red plume, the raw ingredients its inner bacteria need, the things they must have to do their chemistry, and it carries those ingredients down into the trophosome and feeds them to its private population of 1 billion microscopic gardeners. And the bacteria, fed and sheltered and protected from all the dangers of the open water, do what they do best. They take the poison and the chemistry of the vent and they build living matter out of it. And a steady share of that living matter, that freshly made food, flows back into the body of the worm. The worm feeds the bacteria and the bacteria feed the worm, and between the two of them they make a complete creature that needs no mouth and no gut at all. It is one of the most intimate partnerships in all of nature, two utterly different forms of life so woven together that you cannot really say where one ends and the other begins. The worm is, in a sense, a vessel built to carry a garden, and the garden, in return, builds the worm. This is the kind of arrangement that biologists call symbiosis, two living things bound together in a partnership so close that they depend on each other to survive. You have heard of gentler versions of it, perhaps, like the flower and the bee. But there is nothing in your everyday world quite as total as this. The tube worm does not merely cooperate with its bacteria, it surrenders to them. It gives up the whole apparatus of eating, the mouth and the gut and all the machinery of digestion that almost every animal carries, and it hands the entire job of feeding over to the microbes it shelters within. It is a bargain struck in the dark, sealed somewhere deep in the evolutionary past, and it works so well that the worm can grow taller than a person in less than 2 years on nothing but the labor of the tiny lives it keeps inside its body. Think of that the next time you sit down to a meal. There is an animal at the bottom of the sea that has never once taken a bite of anything and grows faster than almost any creature in the ocean. But the trophosome and its billion bacteria are only half of the impossible thing about this worm, because to feed the bacteria, the worm has to solve a problem that should, by all the rules of biology, be unsolvable. And the solution is hidden in that vivid red plume, the part of the worm that reaches up out of the tube and into the warm vent water, the part that glows like blood against the dark rock. Why is the plume red? It is red for the very same reason your own blood is red. It is full of hemoglobin, the iron-rich molecule that carries oxygen through the bodies of so many living things, including yours. When you take a breath, the hemoglobin in your blood reaches out and grabs the oxygen from the air in your lungs and ferries it all through your body to where it is needed. The tube worm's plume is rich with hemoglobin too, and it reaches up into the water to gather what its inner bacteria require. And here is where the trouble begins, the trouble that should have made this whole way of life impossible. The bacteria in the trophosome need two things to do their work. They need oxygen, and they need hydrogen sulfide, the rotten egg poison, the fuel of their chemistry. The worm has to gather both of these from the vent water and carry both of them together down through its body to the waiting garden inside. And that is exactly where the rules say it cannot be done, because hydrogen sulfide is not just a poison to the worm's tissues in the ordinary sense. It has a particular vicious habit. Hydrogen sulfide attacks hemoglobin. It interferes with the very molecule that carries oxygen. In an ordinary creature, in you for instance, if hydrogen sulfide got into your blood in any quantity, it would jam the works, it would block your hemoglobin from carrying oxygen at all, it would shut down the whole delivery system your life depends on. The poison and the oxygen carrier are enemies. You cannot, by the normal rules, ask one molecule of hemoglobin to carry both oxygen and sulfide at the same time, because the sulfide ruins the carrying of the oxygen. It is like asking a single bucket to hold both water and fire. And yet the tube worm does it. Its blood carries oxygen and lethal hydrogen sulfide at the same moment, side by side, all the way down to the trophosome, without either one of them poisoning the worm and without the sulfide wrecking the worm's ability to carry oxygen. For a long time, this looked like it simply should not be possible, and the people who studied it had to work out how on earth the creature managed it. The answer, uncovered by careful science, is that the worm's hemoglobin is not like yours. It is a special, modified version evolved over a very long time to do something yours cannot. The worm's hemoglobin has separate places on it, separate sites, one set for grabbing oxygen and an entirely different set for grabbing hydrogen sulfide. It holds the two of them apart. It carries the oxygen in one set of hands and it carries the deadly sulfide in another, completely separate set of hands, so that the poison never gets the chance to interfere with the oxygen at all and never gets loose in the worm's own body to do its usual damage. The sulfide is bound up safely, locked to its own special handles on the hemoglobin, ferried quietly and harmlessly down to the bacteria that crave it, where it is finally handed over to be used as fuel. So the worm's red blood does something that in your body or in almost any other would be a death sentence. It transports a poison and the breath of life together in the same fluid, in the same molecule, without harm. It is biochemistry that on paper should not work, and the only reason we know it does work is that the worm is down there right now in its millions, swaying in the dark, very much alive. The plume is red because it is full of this remarkable blood, this blood that has solved a problem the rest of us never even have to face, and reaching up into the warm, poisoned water to gather the very things that would kill anything else, so that the garden inside the worm can keep on turning death into life. When you lay all of it side by side, the full strangeness of this single creature begins to settle over you like a weight, a soft one, the kind that presses you gently down towards sleep rather than keeping you awake. Here is an animal with no mouth and no gut, which has given up eating entirely. Here is an animal that grows to the length of a person, faster than almost anything in the ocean, on food it manufactures inside its own body. Here is an animal that keeps 1 billion microscopic farmers in a special organ behind its skin and feeds them, and is fed by them in return in a partnership so deep that neither one could live without the other. And here is an animal whose blood carries a poison and a breath together, in defiance of everything we understand about how blood is supposed to work, all so that it can deliver rotten egg gas to the garden in its belly. Every single thing about it breaks a rule we thought was unbreakable, and it is not rare, and it is not struggling on the edge of survival. It is abundant, it thrives, it builds whole swaying forests of itself around the warm cracks in the floor of the sea. And remember how we found it. We were not even looking. A submarine full of geologists hunting for warm water and interesting rock stumbled across an entire living world that the science of the day said could not exist, fed by a process nobody had properly imagined, built on a poison, thriving in eternal darkness, 2.5 kilometers down. They went looking for the heat of the planet, and they found life living on it. They had no biologist with them, and they had to keep their impossible specimens in vodka because nobody had thought to pack for the discovery of a brand new way of being alive. That single dive in February of 1977 on the Galápagos Rift quietly rewrote one of the deepest assumptions human beings had ever held about life. The sun, we had always believed, feeds everything, and it turned out that the sun feeds almost everything, but not quite all. There is another foundation under the living world, an older and darker one, where the energy comes not from light, but from the raw chemistry of the planet itself, leaking out through the cracks in the seabed. And the tube worm, with its missing mouth and its inner garden and its impossible blood, is the strange and beautiful face of that other foundation. But the giant tube worm, as wonderful as it is only the first creature in a much longer parade, because once scientists knew where to look, once they understood that life could build itself around these hot cracks in the deep, they began to find other animals down there, around vents all over the world, and each one stranger than the last. There is a worm that lives with its tail baking in water hot enough to begin cooking it, while its head stays cool across the span of just a few centimeters of its own body. There is a snail that wears armor made of iron, the only animal on Earth that does such a thing, now slipping toward the edge of extinction. There is a blind shrimp that seems to see heat itself, and a crab that grows a garden on its own arms. And beneath all of these creatures lies a question far larger than any of them, a quiet and enormous question that those first geologists could never have guessed they were diving toward. Because if life can build itself here, in the dark, on poison, with no help from the sun at all, then perhaps the dark is not where life merely survives. Perhaps the dark down here at the bottom of the sea is where life first began. And so the parade continues, deeper and stranger with each step, further from anything our intuitions about life would lead us to expect. There is a worm called Alvinella pompejana, and it lives on the walls of the black smokers themselves, in the hottest animal habitat ever formally measured on this planet. Scientists named it the Pompeii worm, a name that carries its own quiet weight, because Pompeii is a word that means buried alive by heat, a city pressed under volcanic ash, and the Pompeii worm lives in something analogous, a life conducted at the extreme margin where biology and geology blur into each other and ordinary rules cease to apply. The worm builds a papery tube directly onto the wall of a hydrothermal chimney. It lives inside that tube, head pointing outward, tail pressed back toward the superheated rock, and what this means in practice is almost difficult to hold in the mind. The tail of the Pompeii worm sits in water that reaches roughly 80 degrees Celsius. To understand 80 degrees, think of a cup of tea made too hot to drink, a cup you must set down and wait on. That is 80 degrees. And it is the temperature at which the tail of this animal lives routinely as a matter of its ordinary existence. Studies led by Charles Kairei and colleagues, published in the late 1990s, documented these temperatures with probes inserted directly into the worm's tube, and the measurements remained among the highest tolerated by any multicellular animal on record. But here is the thing that makes the Pompeii worm genuinely astonishing, that lifts it from merely extreme to architecturally improbable. The worm is only a few centimeters long, and while its tail bakes at 80 degrees, its head, protruding from the open end of the tube, sits in the ambient water outside, which hovers at around 22 degrees Celsius. 22 degrees. The temperature of a comfortable room, of a mild spring afternoon. Across the span of a few centimeters of its own body, the Pompeii worm sustains a thermal gradient of nearly 60 degrees. It is, in the most literal possible sense, an animal with one end in a sauna and the other end in a temperate garden, simultaneously, with nothing between them but its own biology. No other animal on Earth is known to endure anything close to this. The record belongs, without apparent competition, to a polychaete worm on a hot crack in the Pacific seafloor, 2.5 kilometers below the surface of the ocean, in total darkness, where geologists went looking for mineral formations, and instead found this. How does it survive? That question does not have a complete answer, even now. What is known is that the Pompeii worm is not alone in its tube. It carries a dense coating of bacteria on its back, a kind of living fleece, filamentous microorganisms that carpet the worm's dorsal surface in a pale, hairy layer. The bacteria are thought to provide some degree of thermal insulation, a biological buffer between the worm's skin and the most extreme heat of the surrounding fluid. They may also, as with so many creatures at these vents, provide nutrition, another version of the partnership that runs through all of vent life like a single thread, the animal providing shelter and transport, the bacteria providing metabolic gifts in return. The exact physiology of how the Pompeii worm's cells tolerate the steepest thermal gradient endured by any known animal remains, in large measure, genuinely unresolved. The worm keeps its secret. What we can say is that the proteins in its body, the enzymes that carry out its chemistry, must function across an enormous temperature range within the same organism at the same moment. In most animals, proteins are tuned to a narrow thermal window. Enzymes that work well at 20 degrees will often unfold and fail at 60. The Pompeii worm appears to have solved this, though the molecular details of how are still being worked out. It is the kind of solution that, once you understand what problem it is solving, makes you stop and look at the creature with something close to reverence, because there is engineering in that small body that we do not yet fully understand. There is a kind of comfort, perhaps, in not fully understanding, in sitting with the Pompeii worm and allowing it to remain strange, to remain a question with a partial answer, something the ocean floor has kept to itself. The deep sea does not surrender its logic all at once. It releases pieces slowly, the way a very old place releases its history, one excavation at a time. And now consider the snail. If the Pompeii worm represents the edge of thermal tolerance, a single biological variable pushed past what seemed possible, then Chrysomallon squamiferum, the creature known informally as the scaly-foot snail, represents something altogether more comprehensive in its impossibility. It did not merely push one variable past its limit, it rewrote the periodic table of animal armor. The scaly-foot snail was found at a vent field in the Indian Ocean called the Kairei field, a site on the central Indian ridge where the seafloor cracks and heat escapes in the usual vent manner. It was described formally in 2003, a study led by Anders Warren and colleagues, and what the description revealed was something that, in the entire catalogue of life on Earth, before or since, had never been seen. The snail has a shell, as snails do. But this shell is not made only of the calcium carbonate that shells are normally made of. It is armored with a layer of iron sulfide. The mineral incorporates greigite, an iron sulfide compound, into the outermost layer of the shell in a way that gives it a metallic appearance, dark and dense, more like something forged than grown. And it is not only the shell. The scales, the flat plates that cover the sides of the snail's foot, the muscular pad it uses to move, those scales are armored too. They are called sclerites, and they are coated in the same iron sulfide minerals, dozens of overlapping plates running along each side like a suit of chainmail, like the scales of an animal that evolved in a foundry. Some people who write about this snail call it the sea pangolin. It is an apt name. The pangolin is the only mammal that covers itself in scales made of keratin, a protein armor. The scaly-foot snail is the only animal of any kind known to incorporate iron sulfide minerals into its body armor. In the whole of the animal kingdom, in the entire sweep of evolution on this planet, this appears to be a singular invention. Iron sulfide, the same family of mineral compounds that precipitates out of black smoke of fluid and builds the chimney walls. The snail has taken the chemistry of its environment, the mineral signature of the vent itself, and woven it into its body. It has armored itself in the geology of its own home. The obvious question, and a good one to sit with for a moment, is why? Why go to the metabolic expense of building armor from iron sulfide? The deep sea is not without predators. Crustaceans with crushing claws, fish with pressure-hardened jaws, animals that have evolved to eat whatever they can catch in a place where food is always scarce. The iron armor offers genuine mechanical protection, a hardness and a layered structure that studies showed absorbed force in ways that simple calcium carbonate could not. Later analysis of the scaly-foot snail's shell structure found that the layered system, the outer iron sulfide, a middle organic layer, and an inner calcium carbonate distributed stress across the layers in a way that prevented cracking from propagating. It is, in engineering terms, a composite armor system, and it evolved at a volcanic vent in the Indian Ocean, in an animal roughly the size of a large grape. But the snail's strangeness does not end with its exterior. Inside there is something else. The snail's heart is disproportionately enormous. Measured against its body volume, its heart takes up roughly 4% of the whole. In a human being, the heart is less than 0.5% of body volume. The scaly-foot snail's heart, scaled to its size, would be nearly 10 times as large as a human heart relative to the body around it. And the reason for this is by now familiar in the logic of vent life. The snail carries in an enlarged esophageal gland a vast population of symbiotic, chemosynthetic bacteria. As with Riftia and its trophosome, the bacteria are the nutritional engine, the internal partners that feed the animal by fixing carbon from the chemistry of the vent. And to keep those bacteria supplied with oxygen and sulfide in sufficient quantities, the snail needs to push enormous volumes of fluid through its body. The great heart is not a luxury, it is infrastructure. It is the industrial pump that keeps a bacterial city alive inside a creature smaller than your fist. Studies examining this heart, including work by Chong Chen and colleagues published in 2015, describe the cardiovascular system of the scaly-foot snail in detail and noted that in all likelihood, the organ's size reflects the metabolic demands of the bacterial community within. The snail and its bacteria are not merely in partnership. They are in a relationship so deep that the snail's anatomy has reorganized itself around the needs of the microbes it carries. The heart beats, the fluid moves, and somewhere inside that small armored body, bacteria are turning the chemistry of volcanic vents into the food that keeps the whole structure alive. And that structure, for all its ingenuity, is precarious because the scaly-foot snail does not live everywhere. It lives at a handful of known hydrothermal vent fields in the Indian Ocean, patches of seafloor only a few hundred meters across, warm and chemically active and biologically dense, surrounded by the vast, cold, essentially barren abyssal plain. It is not a numerous animal across the broad ocean. It is an animal adapted to one of the rarest microhabitats on Earth, a specific combination of chemistry and heat and mineral flux that occurs only at certain points where the tectonic plates beneath the Indian Ocean pull apart and let the planet breathe. In 2019, the International Union for Conservation of Nature placed Chrysomallon squamiferum on its red list of threatened species. The status given was endangered. It was the first deep-sea hydrothermal vent species to receive a formal conservation listing of that kind. The threat, notably, is not pollution in the conventional sense, not coastal runoff or plastic or the familiar catalogue of shallow water harms. The threat is mining. The polymetallic sulfide deposits that build up around hydrothermal vents, all that precipitated metal, the copper and zinc and gold and rare elements that the Earth is slowly concentrating there, have begun to attract commercial interest. The technology to mine at those depths is being developed. And the places that would be most valuable to mine are precisely the places where these creatures live, the vent fields themselves, the few hundred square meters of active seafloor where the chemistry flows and the iron snail makes its improbable home. There is something to sit with in that. An animal that took millions of years to evolve, the only iron sulfide armor in the history of animal life, that reorganized its own heart to feed an internal bacterial civilization, that made its home in one of the rarest and most chemically extraordinary environments on Earth, now listed as endangered because the minerals it built its house from are valuable to us. The geology that made the snail possible is the same geology that puts it at risk. It is the kind of irony that the deep sea offers without comment, without drama, simply as a fact. Let the snail rest for a moment in its iron house. Let it pulse its enormous heart slowly in the dark. There are others in this parade. The yeti crab, which belongs to a genus called kiwa, was described in 2005, discovered on the Pacific Antarctic Ridge, a stretch of seafloor between the southern tip of South America and Antarctica, where the plates pull apart and vents form in some of the most remote water on Earth. It is a pale, almost ghostly crustacean, and its most immediately striking feature is that its claws and the walking legs around them are covered in dense, silky setae, fine filaments that in photographs give the animal a distinctly furry appearance, like a crab that wandered in from the wrong continent. The name yeti crab was not accidental. The discoverers, Emmanuel McPherson and colleagues, were making explicit the quality of the animal's strangeness, its belonging to a category of thing that should not quite exist, but evidently does. What the yeti crab does with its fur is, when you understand it, one of the more elegant behavioral solutions in all of vent biology. The setae are not decorative and they are not simply sensory. They are a farm. Living on those filaments, cultivating in the spaces between them, are colonies of chemosynthetic bacteria. The crab grows its food supply on its own body, and more than that, it actively manages the cultivation. Studies have observed yeti crabs performing a characteristic slow wave of the claws, a back-and-forth motion that brings the bacteria-laden setae through the plume of warm, chemical-rich vent fluid. Hydrogen sulfide and the other compounds the bacteria need are carried in that fluid. By waving its arms through the vent flow, the crab is irrigating its farm, delivering the metabolic raw materials to its bacteria the way a gardener directs water to roots. Then, when the bacteria have grown and multiplied on the setae, the crab grazes them, scraping the crop from its own claws with a specialized comb structure near its mouth. It grows its food on its arms and then harvests it with its face. In the permanent darkness of the deep Pacific, this is the solution that evolution arrived at. Become your own garden, carry your own bacteria, tend them with your claws. The elegance here lies in the problem being solved. Food is scarce in the deep sea. The water column above carries very little nutrition by the time anything settles to the abyssal floor. Vent ecosystems are rich compared to the surrounding seafloor, but even there competition is intense. An animal that can internalize its food production, that does not have to hunt or scavenge but simply grows, is solving a resource problem in a way that is as efficient as anything evolution has produced. Riftia solved it by moving the bacteria inside its body entirely. The yeti crab found a different answer, keeping them external, on the surfaces of its claws, where they can be irrigated by the very environment the crab inhabits. Different architecture, same logic, chemistry to food without sunlight, in the dark. And now the shrimp. In the Atlantic Ocean, on the vents of the mid-Atlantic ridge, the dominant animal is not a tube worm. The communities there are different from the Pacific ones, a divergence in vent fauna between ocean basins that reflects how isolated these oasis ecosystems are, how the vast, cold stretches between vents act as barriers that prevent mixing, allowing evolution to find different solutions on different sides of the world. The Pacific vent fields are dominated by the creatures we have been meeting, the Riftia tube worms, the Pompeii worms clinging to the chimneys, the pale crabs and anemones clustered at the margins. The Atlantic fields, particularly the thermal vents along the mid-Atlantic ridge, are dominated by shrimp. Rimicaris exoculata. The name means, roughly, the cave-dwelling shrimp without eyes, and this is accurate as far as it goes. The conventional compound eyes that most shrimp possess, the clustered lenses arranged to capture light and form images, are absent in Rimicaris. The ocean at the depth these shrimp inhabit is lightless in any conventional sense. 2.5 kilometers of water column stands between the surface and the seafloor, and essentially no visible light penetrates to that depth. Standard eyes in that darkness are an investment with no return. Evolution disposed of them. But the story does not stop there, because the shrimp is not in any simple sense blind. On the back of the Rimicaris exoculata's carapace, the shell covering its main body, there is a modified photoreceptor organ. It is not an eye in the way we usually understand eyes. It does not form images. It does not allow the shrimp to see shapes or movement or color. What it does, or what the most well-supported hypothesis suggests it does, is detect extremely faint light at wavelengths consistent with thermal radiation, the faint glow emitted by very hot matter. Cynthia Van Dover and colleagues described this organ and its likely function in 1989, noting that water heated to several hundred degrees emits a small but measurable quantity of light at the far edge of the visible spectrum. Light that would be invisible to human eyes even if we could somehow be present at those depths, but which falls within a range that the photoreceptor of Rimicaris may be sensitive to. What this means, functionally, is that the shrimp may be able to navigate toward or away from the vent fluid itself, using the thermal glow of the superheated water as a navigation signal. The vent is, in effect, a beacon, not a light beacon, not in the human sense, a heat beacon, a thermal beacon, radiating a signature at the boundary between physics and perception that the shrimp has evolved to read. Why would this matter? Because the vent fluid that emerges from a black smoker at 350 to 400 degrees Celsius will kill a shrimp instantly. The organisms that cluster around vents do not sit in the hottest fluid. They crowd the margins, the zones where superheated vent water has mixed with cold seawater enough to be warm but not lethal, where chemical gradients are steep and food is dense but the temperature is survivable. Rimicaris, potentially, uses its modified back eye to locate those margins, to find the edge of the danger zone and stay just outside it, to know when it has drifted too close to the lethal heat by the increasing intensity of the thermal signal pressing through its carapace. It is navigation by a sense we do not have and cannot easily imagine in a world without light, reading geography by the physics of heat radiation at the bottom of the Atlantic Ocean. Rimicaris exoculata swarms around those vents in enormous numbers, a dense carpet of pale bodies jostling for position in the current, each one orienting itself by the faint warmth pressing through the darkness. They carry chemosynthetic bacteria in their gill chambers, another iteration of the vent partnership, and they are the dominant consumers at many mid-Atlantic vent sites, the animal that fills the ecological niche that tube worms fill in the Pacific. Same vent, same chemistry, a completely different animal. Evolution, facing the same problem in a different ocean, drafted a different answer. Hold all of these creatures in mind for a moment, not individually but as a company. The Pompeii worm with its gradient of 60 degrees across a few centimeters of living tissue. The scaly-foot snail pressing its massive heart to supply its bacterial tenants, sealed in iron armor, newly classified as endangered. The yeti crab waving its furry claws through volcanic fluid to irrigate a garden growing on its own body. The eyeless Atlantic shrimp reading the glow of hot rock through a modified organ on its back, swarming in the permanent dark of the mid-Atlantic ridge. And behind all of them, the one we met first, Riftia pachyptila, the giant tube worm, its red plume extended 2 meters into the water column, carrying blood that holds oxygen and poison in the same molecule without choosing between them, feeding on 1 billion bacteria per gram of its own interior tissue, growing its full length in under 2 years on a vent that might close tomorrow. What is the category these animals belong to? They are not extremophiles in the narrow sense that word sometimes implies, merely tolerating an unpleasant environment the way a cactus tolerates a desert. These animals are not merely enduring the deep vent environment, they are built from it. Their biochemistry is an expression of volcanic chemistry. Their partnerships with bacteria are not adaptations grafted onto an already functional body plan. The bacteria are the plan. The animal bodies, in many cases, exist partly as housing, as plumbing, as delivery infrastructure for the microbial chemistry that does the actual work of nutrition. The animal provides the superstructure, the bacteria provide the engine. And this is not a marginal or unusual strategy, it is the dominant strategy at these vents. Look at almost any abundant animal at a hydrothermal vent, and you will find, upon looking closely, symbiotic, chemosynthetic bacteria somewhere in the picture. In the trophosome of Riftia, in the esophageal gland of the scaly-foot snail, in the gill chambers of Rimicaris, on the setae of the yeti crab's claws. The bacteria are not peripheral. They are the common thread running through all of vent life the way chlorophyll runs through the life of a forest. The forest runs on sunlight through chlorophyll. The vent runs on volcanic chemistry through bacteria. And the bacteria themselves, those chemosynthetic archaea and microbes that oxidize hydrogen sulfide and fix carbon in the dark, are not a specialized curiosity. They are ancient. Their metabolic pathways, the biochemical routes they use to turn chemical energy into biological molecules, trace back to some of the earliest chapters of life on Earth. The question that was always implicit in the 1977 Alvin discovery, the question that geologists dove toward without knowing it, is whether the deep vent environment is not merely a home that ancient life found, but the place where life first assembled itself. Whether these bacteria and the strange animals that depend on them are not exceptions to the main story of life, but a remnant of it, a living fossil of the beginning. That question requires a different kind of patience to approach. It requires looking not just at the animals clustering around today's vents, but at the structure of vents themselves, at the chemistry they produce, and at what the chemistry of a vent looks like when you hold it next to the chemistry that every cell in your own body uses to stay alive. Consider what you are in the most basic chemical sense. Every cell in your body runs on a molecule called ATP, adenosine triphosphate, the universal energy currency of life, the thing that powers every protein motor, every chemical reaction, every heartbeat. And how do your cells make ATP? They make it by running a proton gradient across a membrane. Inside the mitochondria of your cells, there are membranes studded with molecular machines, and those machines drive protons, hydrogen ions, across the membrane from one side to the other, building up a difference in concentration, a gradient. Then, as those protons flow back down the gradient through a different molecular machine, the flow is harnessed to build ATP. It is, in miniature, a kind of waterfall mill, the flow of protons down a gradient turning a wheel that does chemical work. This mechanism, chemiosmosis, is universal. It is found in every domain of life, in bacteria and archaea and every cell of every plant and animal and fungus that has ever lived. Peter Mitchell worked out the essential principle and received the Nobel Prize in Chemistry for it in 1978. And the universality of the mechanism is, to the scientists who think carefully about the origin of life, not just an interesting biochemical fact, it is a clue. When something is universal to all known life, the simplest explanation is that it was present in the common ancestor of all known life, which means that before there were mitochondria, before there were complex cells, before there were bacteria of the kind we recognize today, something was already running on proton gradients. The mechanism that keeps you alive and that kept every living thing that has ever existed alive is one of the oldest things in biology. It predates almost everything else we know about life. And now consider what an alkaline hydrothermal vent does. Not the dramatic black smoker with its superheated fluid and its metal sulfide chimneys, but a different, quieter kind of vent. A vent driven not by a magma chamber directly heating water, but by a chemical reaction between seawater and a rock called peridotite. When seawater percolates down into certain kinds of ocean floor rock, the minerals in that rock react with the water in a process called serpentinization. The rock absorbs the water, expands slightly, and in the process releases heat and generates, among other things, molecular hydrogen. The fluid that comes back up through the rock is warm, not scalding, somewhere between 40 and 90 degrees Celsius, rather than several hundred, and it is highly alkaline, with a pH that can reach 10 or 11, while the ancient ocean it vents into would have been mildly acidic. The vent fluid is alkaline, the surrounding water is acidic. Where they meet at the mineral walls of the vent structures, there is a natural proton gradient, not a manufactured one, not a biological one, a geological one. Protons flow across those thin mineral walls the way they flow across the membranes inside your mitochondria, for the same reason, because there is a difference in concentration on either side. And the mineral walls of alkaline vents are porous, riddled with tiny compartments, chambers a fraction of a millimeter across, microscopic spaces enclosed by thin mineral barriers that are permeable to certain molecules in the same way biological membranes are permeable to certain molecules. A natural proton gradient. A steady supply of hydrogen, one of the most useful chemical energy sources biology knows. Microscopic mineral compartments that enclose chemistry the way cells enclose chemistry. These are the features that a group of scientists, including Michael Russell and William Martin and Nick Lane, identified as potentially providing the starting conditions for life itself. Not randomly, not all at once, but in a way that geologically generates, for free, the same basic physical situation that every living cell has been reconstructing biochemically ever since. This is the alkaline hydrothermal vent hypothesis for the origin of life. It is a hypothesis and it is explicitly debated, and returning to that distinction will be important. But the logic of it, before any verdict is reached, is worth moving through slowly because it is one of the more quietly remarkable ideas in modern science. The proposal is not simply that alkaline vents are hospitable to early life. The proposal is that the specific mechanism all life uses to generate energy, the proton gradient and the chemiosmotic wheel, is something that vents of this kind produce geologically, spontaneously, without biological machinery, driven purely by the chemistry of rock and water. If life's energy mechanism was not invented by life but was instead inherited from the environment, then the question of how life started becomes considerably less bewildering. You do not have to imagine a cell appearing from nothing, somehow already equipped with membranes and proton pumps and ATP machinery. You can imagine instead chemistry happening in a place that was already producing the right gradients, chemistry that over time became better and better at using those gradients, until what was happening in those mineral compartments was no longer just chemistry, but the very first thing we might call biology. And so before turning toward that alkaline quietness and what it might mean, it is worth understanding the other kind of vent the kind that announced itself first, the kind that defined the word hydrothermal for the generation of scientists who came after 1977. Because the two types are not simply variations on a theme, they are almost opposites, and the contrast between them is part of what makes the origin of life argument so interesting. The black smoker is, in the most literal sense, a chimney. It is a structure rising from the seafloor, sometimes a meter tall, sometimes 10, sometimes much more, built entirely from minerals that were dissolved in superheated fluid and then precipitated out, crystallized, hardened, stacked, the moment that fluid met cold seawater. The process is continuous. Hot fluid flows up through the interior of the chimney, and at the lip, where it meets the surrounding ocean, minerals drop out of solution and the structure grows, not quickly by human standards, but measurably, steadily, in geological terms, almost urgently. And the fluid itself, when it exits, carries temperatures that are genuinely difficult to hold in the mind. Around 350 degrees Celsius, some vents pushing toward 400. These are temperatures at which lead melts, temperatures at which many metals would begin to lose their structural integrity, temperatures far beyond any ordinary biological threshold, beyond any chemical process most people have ever encountered outside of a furnace or a flame. And yet the water does not boil. This is the pressure. The weight of 2.5 kilometers of ocean pressing down on that water with a force roughly 250 times the pressure of the atmosphere at sea level holds the molecules together, prevents the transition to steam, keeps the fluid in a superheated liquid state that has no real equivalent at the surface of the Earth. It is, in a sense, water that should not exist at that temperature, water that the sheer mass of the ocean above will not permit to become anything other than liquid, no matter how hot it gets. And that liquid, at 400 degrees, shot through with dissolved metals, copper, zinc, iron, manganese, and carrying hydrogen sulfide in concentrations that would be lethal to most organisms within seconds, pours out of the tip of the chimney and hits seawater at around 2 degrees Celsius. What happens in that collision is immediate and dramatic. The metals drop out, the sulfides precipitate. Dark particles form so rapidly and in such abundance that the vent appears to exhale black smoke, a dense, billowing, roiling column of mineral particles rising into the water column above, catching whatever faint bioluminescence exists in the surrounding darkness and appearing for all the world like the stack of a ship's engine or the exhaust of some underground industrial process running without any human design or oversight. Hence the name. The black smoker does not actually smoke, but the illusion is so complete and so persistent that the word stuck immediately and has never been dislodged. And because the precipitation is constant, because the minerals keep falling out of solution at the chimney's lip, with every moment the vent flows, the chimney grows, the structure accreates around itself, mineral on mineral, crystals interlocking, sulfides bonding to sulfides, and the whole tower rises as years and decades pass. Most chimneys are modest, a few meters, perhaps 10 or 15, structures you could walk past on a cliff face without feeling them to be particularly extraordinary. But some grow to dimensions that feel almost architectural. On the Juan de Fuca Ridge, the sprawling tectonic boundary that runs along the seafloor off the northwest coast of North America, one black smoker chimney grew to a height of roughly 45 meters, 15 stories by the common measure. A structure taller than many buildings, built entirely by precipitation, by the automatic and unguided physics of hot fluid meeting cold water. It was given a name, Godzilla, and it grew and it was documented, and researchers returned to it across multiple expeditions. And then at some point in its growth, the structural weight exceeded what the base could support, and it collapsed, as all chimneys do eventually. The whole tower, folding back to the seafloor, to become the raw material for new growth if the vent below it continued to flow. The lifespans of black smokers are, by geological reckoning, brief. A vent field driven by the heat of magma close to the surface is dependent on that magma maintaining its position and its heat. When the tectonic system shifts, when the magma reservoir cools or migrates, the vent field above it can go quiet within years, sometimes within decades. The hydrothermal plumbing that directed the fluid upward seals off, or fills in, or collapses, and the entire ecosystem that had built itself around the heat and the chemistry of that particular vent, the tube worms, the clams, the shrimp, the bacteria coating every available surface, dies. Not immediately, but as the fluid slows and cools, as the hydrogen sulfide diminishes, as the chemical energy that powered the food web at its base fades, the system unravels. What remains eventually is the chimney structure itself, or what is left of it, resting on the sea floor as a kind of fossil, a record that something was here, that water moved here, that life was here, though the life itself is now gone. This impermanence is one of the things that makes vent ecology so striking when you think about it carefully. These are not stable environments by any ordinary measure. They are oases, genuine oases, patches of warmth and chemical richness in an otherwise featureless and frigid plain, and they arise and they vanish on timescales that are short, even by the standards of organisms that live only a few years. And yet the life finds them, and not only finds them, but colonizes them with a speed and thoroughness that is itself one of the more astonishing documented facts of vent biology. In April of 1991, a volcanic eruption along a stretch of the East Pacific Rise near 9 degrees north latitude sterilized a portion of the seafloor that had, until that eruption, hosted a thriving vent community. The lava came up fast, as it does in these shallow submarine eruptions, and it buried and cooked everything in its path. When researchers descended in Alvin shortly after, they found what has been described as a sea floor covered in what looked like cooked organic material. Pale, collapsed, dead. The worms were gone. The clams were gone. The microbial mats were gone. The lava had reset the system completely, erasing what had been there and presenting the seafloor as a blank, still cooling surface, waiting for something to happen. The team returned, and what they found in the months and years that followed, documented through a series of return visits that became one of the most detailed ecological succession studies ever conducted in the deep sea, was a recolonization so rapid it seemed almost implausible. Within months, microbial mats had begun forming on the new lava surface, feeding on whatever hydrogen sulfide was seeping through. Within roughly a year to 2 years, larger organisms had appeared. The Riftia tube worms, those 2-meter-long animals with no mouths, those animals whose larvae must somehow make the crossing from other vent communities elsewhere, had arrived and were already establishing themselves, growing at the rates documented by Richard Lutz and his colleagues. Rates that made them among the fastest growing marine invertebrates known, so that within that first year or two they were already visible, already recognizable, already forming the beginnings of the dense waving communities that would eventually repopulate the site. Consider what that crossing requires. A Riftia larva is small. It carries yolk reserves for only a limited time before it must either find a suitable surface and settle or exhaust its resources and die, and it has no map. It has no guidance system beyond whatever chemical gradients it can detect in the water around it, whatever faint traces of hydrogen sulfide or heat might diffuse up from a vent into the water column above. The seafloor between vent fields is not a gradual transition. It is largely lifeless. The abyssal plain, the flat and frigid sediment floor that stretches between these scattered oases, is one of the most food-poor environments on the planet. There is almost nothing there. Particles of organic material drift down slowly from the sunlit ocean far above, marine snow, the constant gentle rain of dead plankton and fecal pellets and fragments. And that is essentially the energy budget for the vast expanses between vents. A Riftia lava crossing those expanses is crossing a desert, an immense and featureless darkness, and it must do so within the window its yolk reserves allow, and it must find, in that darkness, the chemical signature of a vent. That it does find them, that vent communities reconstitute themselves on the timescales documented at 9 degrees north, tells you something about how strong the pressure to exploit these chemical environments must be. The larvae are there in the water column, in numbers sufficient that enough of them, drifting with currents, make the crossing and find the new surface. The system is tuned for exactly this kind of rapid, repeated colonization, because the environments themselves are ephemeral enough that an organism which cannot recolonize quickly enough will simply go extinct when its home vent goes dark. And this is the ecological texture of the hydrothermal vent world, a world of scattered islands, each burning briefly and intensely with chemical energy, each drawing from a shared larval pool drifting in the water between them, each distinct in its precise chemistry and its precise fauna while sharing the underlying logic of chemosynthesis at its base. Across different ocean basins, the communities that build around vents are not identical. The Pacific vent fields, like those of the East Pacific Rise and the Juan de Fuca Ridge, are dominated by the organisms that became familiar from 1977 onward. The Riftia tube worms forming dense, red-plumed forests, the giant white clams and mussels, the various crabs and fish that prey on them, and everywhere the microbial mats forming the foundation of it all. The Pacific fauna has its own character, built over the evolutionary time available since the Pacific vents were first colonized, and it reflects a community that has been shaped by those particular vent chemistries and those particular geological rhythms. Cross to the Atlantic, and the fauna shifts. The mid-Atlantic ridge running down the center of the Atlantic Ocean, like the seam of a basketball, hosts its own vent fields, and they are chemically similar in broad strokes: hot fluid, hydrogen sulfide, metal sulfides precipitating. But the biological community is strikingly different. Here, the emblematic animal is not the Riftia tube worm, but a shrimp. Rimicaris exoculata, the vent shrimp of the mid-Atlantic, swarms around its vents in densities that are almost difficult to picture. Thousands and thousands of individuals pressing close to the hot fluid, jostling, moving, the entire surface of a vent structure sometimes appearing to be entirely composed of shrimp in motion. A creature whose Latin name acknowledges what is immediately obvious about it: exoculata, from without eyes. It has no eyes in the conventional sense, no compound eyes, no light-focused visual system of the kind you would find in a shrimp living anywhere near the ocean's surface. What it has instead is stranger, and for a long time it puzzled researchers considerably. On its back, beneath a transparent patch of carapace, Rimicaris has a large, flat, light-sensitive organ. An organ that has no counterpart in the visual systems of its surface-dwelling relatives. It is not an eye in any usual sense. It cannot form an image. It cannot resolve edges or shapes or movement in the way that an eye does, but it is exquisitely sensitive to light, and the question that followed its discovery, confirmed in 1989 by Cindy Van Dover and colleagues, was, what light? At 2,500 meters depth, in water far below the reach of any sunlight, in a darkness so complete that the surface far above might as well not exist, what was this organ detecting? The answer, when it was worked out, was heat. The fluid exiting a black smoke event at 350 to 400 degrees Celsius is so hot that it radiates, not in visible wavelengths but in the near-infrared, and at the very edge of what might be detectable as extremely faint, reddish light by a sufficiently sensitive organ. The vent itself, the superheated fluid, and the hot chimney walls emits black-body radiation, the kind of faint electromagnetic emission that any object at a high enough temperature will produce, though in this case in quantities so small that even calling it light requires stretching the word. Rimicaris, it seems, can detect this. Its back-mounted organ gives it a faint impression of the vent's presence, enough to orient toward it, enough to maintain its position in the swarm without straying so close that the temperature becomes lethal. It sees heat in the dimmest possible sense at the bottom of the ocean in a world where seeing anything at all is a feat that should by rights be impossible. There is something that stays with you about that adaptation. The shrimp arrived here, evolutionarily, from surface waters where sight is a straightforward proposition, where light comes from above, where eyes are useful, where the visual world is rich with information. And somewhere in the descent, across the generations, sight became useless and then vestigial and then gone, replaced by something more immediately practical, something calibrated not to the sun but to the heat of the sea floor itself. The shrimp does not see the vent the way you see a lamp in a dark room. It registers the vent the way you might register warmth on your skin from a nearby fire. Not an image, not a picture, but a gradient, a direction, a sense of where the heat is coming from. And that is enough. Enough to swarm, enough to feed, enough to survive in an environment where conventional sight would be a metabolic expense with no return. The Atlantic and Pacific vent faunas are sufficiently different that biogeographers have spent considerable effort trying to understand how the separation arose and how the communities are maintained. The deep ocean is not a connected system in the way surface waters are. The mid-ocean ridges that generate vents form barriers as much as pathways. The different ocean basins have different tectonic histories, different ridge spreading rates, and those differences have given rise to communities that, despite sharing the same fundamental chemosynthetic base, look and function differently. This divergence is itself a piece of evidence that vent communities are genuinely isolated from each other in ecologically meaningful ways, that the crossing between vent fields is genuinely difficult, that the larvae that manage it are accomplishing something that most individuals of their kind fail to do. And so the vent world, seen from the outside, is an archipelago, scattered islands of heat and chemistry in an enormous cold plain, each one brief by geological time, each one intensely alive while it lasts, each one drawing on larval resources drifting in the darkness between them, each one eventually failing and going dark, while somewhere else a new crack opens in the seafloor, and the fluid begins to rise and the chemistry begins again, and within months to years, something arrives to claim it. It is a world built on impermanence and on the capacity to find and exploit impermanence faster than the competition, and it has been running in various forms for as long as the seafloor has been producing the tectonic conditions that allow it. Now, the black smoker, for all its drama, the superheated fluid, the 45-meter chimney, the clouds of precipitating metal sulfides, the shrimp swarming in thousands, is not where the origin of life hypothesis centers. The black smoker is too hot, its chemistry too extreme, its pH too acid, its instability too pronounced. An organism trying to get started in the outflow of a black smoker is working against chemistry that destroys organic molecules nearly as fast as they form. The origin of life argument lives somewhere else. It lives in a place discovered much later, and stranger in its own way, and quieter in almost every respect. In the year 2000, a research expedition operating in the Atlantic Ocean made a discovery that added a new and entirely different chapter to the story of hydrothermal vents. The team was working near the Atlantis Massif, a block of ancient oceanic crust rising from the seafloor near the mid-Atlantic ridge, and what they found there was a hydrothermal field unlike anything previously documented. Not black smokers, not the superheated, acid, metal-rich venting that had defined the field since 1977. What they found at the Lost City Hydrothermal Field, as it would come to be called, was white, towering white structures rising from the seafloor, some of them immense, built not from metal sulfides but from carbonate minerals, the same broad family of minerals that makes limestone, that builds seashells, that gives chalk its color. Carbonate structures built by a fundamentally different chemical process, rising in silence from the seafloor in the quiet Atlantic dark. The tallest of these structures, given the name Poseidon, stands approximately 60 meters above the seafloor, 60 meters, higher than the Godzilla chimney that collapsed on the Wanda Fuca Ridge. A structure nearly as tall as some lighthouse towers, nearly as tall as some church steeples, built entirely from carbonate minerals, precipitated by a chemical process happening in the rock beneath it. And unlike Godzilla, unlike most black smoker chimneys, Poseidon and its neighbors at the Lost City Field appear to have been venting and growing for a very long time. The estimates for how long the Lost City system has been active run to more than 30,000 years, and some assessments of the geological setting suggest the system may be older still. Where a typical black smoker field might run for decades before its magmatic heat source migrates or cools, the Lost City Field draws on a different process entirely, a process that does not depend on nearby magma and is therefore not subject to the same kind of abrupt shutdown. That process is serpentinization. The word sounds almost medieval, as though it belongs in an alchemist's notebook rather than a geochemistry paper. But what it describes is a chemical reaction that happens when certain types of rock, specifically the dense iron and magnesium-rich rocks that make up the mantle, the deep layer of the earth beneath the crust, come into contact with water. The Atlantic Massif is made partly of this kind of rock, mantle material, that has been uplifted toward the sea floor by tectonic forces. When seawater percolates down through fractures in this rock, it reacts. The minerals in the rock, olivine and pyroxene, if you want the specific names, absorb the water and are transformed, chemically altered into a new suite of minerals called serpentinite, and in the process of that transformation, heat is released and hydrogen gas is produced. The hydrogen is the important thing, not the heat, though the heat is what drives the fluid circulation that builds the white chimneys above. The hydrogen is what makes Lost City relevant to the question of where life began. Because hydrogen is a reducing agent, a source of chemical energy, a molecule that bacteria and archaea can eat, and in the Lost City system it is produced continuously, without any biological input, driven purely by the chemistry of rock and water, reacting in the darkness of the seafloor. Seawater goes down through cracks, it reacts with mantle rock. Hydrogen comes up along with methane, also produced by the serpentinization chemistry, and the fluid that rises is alkaline rather than acid, warm rather than scalding, carrying a steady and geologically reliable supply of the molecule that vent microbial communities use as their primary energy source. When the fluid reaches the seafloor and vents into the surrounding ocean, which at the time of Lost City's formation billions of years ago would have been acidic, rich in carbon dioxide, a very different ocean from today's, the contrast between the alkaline vent fluid and the acidic seawater creates something. It creates a gradient. A difference in chemistry across the thin mineral walls of the carbonate structures the vent builds. And this gradient is not just a chemical curiosity. It maps with a precision that is striking onto the gradient that every living cell on Earth maintains across its own membrane. The proton gradient, the engine of chemiosmosis. The mechanism that Peter Mitchell described in his work, honored with a Nobel Prize in 1978, the universal mechanism by which cells across all of life's domains harvest chemical energy and convert it into the molecule ATP, the universal currency of biological work. Every time you move a muscle, every time a neuron fires, every time a cell divides or builds a protein or repairs its own membrane, the immediate energy driving that work has, almost always, come through chemiosmosis, through the movement of protons, hydrogen ions, across a membrane from a region of higher concentration to lower, through a protein machine that uses the flow of those protons to link phosphate groups together and build ATP. It is a mechanism so fundamental to life that it operates in your cells and in the cells of a bacterium living in a hot spring in Yellowstone and in the cells of an archaeon living in the sediment of the abyssal plain and in every organism in every environment where life has been found. It is, in the language of evolutionary biology, deeply conserved, preserved across billions of years of evolution, maintained through every branching of the tree of life, because it works so well and runs so deep that abandoning it would be more costly than keeping it regardless of whatever else changed. And the alkaline hydrothermal vent, of the kind found at Lost City, produces a proton gradient across its mineral walls without any biology at all. The alkaline fluid inside the vent pores has a different proton concentration from the acidic seawater on the outside. That difference, that gradient, is geological. It arises from serpentinization chemistry. It is maintained for tens of thousands of years by the slow, steady reaction of rock and water. And the pores of the carbonate mineral structures that the venting fluid builds are small, labyrinthine, interconnected, with walls thin enough that the gradient across them resembles, in its basic physical structure, the membrane of a living cell. This is the foundation of the argument that Michael Russell began developing, and that William Martin and Nick Lane subsequently extended and elaborated. Not simply that alkaline vents could support early life, but that the very energy mechanism life uses, as the proton gradient, the chemiosynthetic machinery, was present geologically before life existed, and that life, rather than inventing this mechanism from scratch, may have simply learned to internalize and improve upon a gradient that its environment was already providing for free. It is worth pausing on that idea because it reframes the origin of life problem in a way that makes it feel, if not easier, then at least less arbitrary. The question of how life started has always faced the challenge of emergence. How do you get from chemistry something as organized and specific as a living cell? One of the most persistent difficulties is the energy problem. Life needs energy to build itself. Building the molecules of life, the nucleic acids, the proteins, the lipids, requires energy. But the machinery that harvests and converts energy is itself made of molecules that had to be built. This looks, from a certain angle, like a circle. You need energy to make the machines, but the machines are what you use to get energy. In the black smoker world, this circle is hard to break. In a vent system where the gradient is already there, already doing chemistry, already driving the flow of protons across a membrane-like mineral wall, in that system the circle becomes, if not broken, then at least loosened. The energy is available before the biology is. The environment provides the gradient. The chemistry, over time, learns to use it. The Lost City field has been venting for more than 30,000 years, and the process that drives it, serpentinization of mantle rock, would have been occurring on the early Earth as long as there was ocean water percolating into exposed mantle material. The early Earth had a great deal of exposed mantle rock and a great deal of ocean water. The conditions for this kind of venting are not exotic or rare in the deep time context of the planet's early history. They are, by the standards of what geology requires to produce them, relatively straightforward. And they would have been happening on some early seafloor for a very long time before any life existed to encounter them. This is the setting that the alkaline vent hypothesis proposes for the emergence of life. Not a warm little pond at the surface, catching sunlight and occasional lightning. The image associated with Darwin's offhand phrase in a letter, the image taken up and developed more rigorously by researchers including Bruce Damer and David Deamer, who have done serious and careful work on the warm pond scenario and who represent a genuine and competing hypothesis that should not be dismissed. Not a volcanic hot spring on land, the kind of environment proposed by Arman Mulkijanian and others, who point to the specific mineral concentrations found in land-based geothermal systems as better matches for the ionic chemistry of living cells. These are competing hypotheses, and they are serious ones, and there is real scientific work on each of them, and the field has not reached consensus and may not reach it soon. The question of where life began is genuinely open. What the alkaline vent hypothesis proposes, among those competing options, is a setting at the bottom of an ancient sea in a field of white carbonate towers built by serpentinization chemistry in pores and channels through which alkaline hydrogen-rich fluid flowed against an acidic, carbon dioxide-rich ocean, creating gradients across thin mineral walls that did for free what a living cell would eventually learn to do with biological machinery. And the hypothesis connects to a remarkable piece of genomic work published in 2016 by Weiss and colleagues in Nature Microbiology, in which researchers attempted to reconstruct the biology of LUCA, the Last Universal Common Ancestor, the organism or population of organisms from which every living thing on Earth today descends. That reconstruction, the Weiss et al. Study, is itself a remarkable kind of archaeology. You cannot dig up LUCA, there is no fossil of it, no preserved cell wall, no bone. What you can do instead is look across the entire span of life as it exists today. Bacteria on one branch, archaea on another, and all the eukaryotes, all the creatures with nucleated cells, including you, on a third. And ask what genes are shared so deeply and so universally across all three domains that they must have been present in the common ancestor before the branches diverged. The logic is like looking at three languages that developed independently for thousands of years and finding the words that are almost identical in all three. Those words were probably in the parent tongue. What Weiss and colleagues found when they ran that comparison across the genomes of organisms from all three domains was approximately 355 gene families that met the criteria for having been present in LUCA. Not 355 individual genes exactly, because genes mutate and drift, but 355 clusters of related genes pointing back toward an ancestral function. And when they asked what those gene families, taken together, implied about how LUCA lived, what it ate, what environments it could tolerate, what chemical reactions it depended on, a picture emerged that is, at minimum, striking. LUCA's inferred biology, as reconstructed from that genomic evidence, pointed toward an organism that depended on hydrogen gas, that fixed carbon dioxide, that required metals as cofactors in its enzymes, that lived in an anaerobic environment, meaning one without free oxygen, and that appeared to be thermophilic, meaning it preferred heat. Thermophilic, dependent on hydrogen. Anaerobic, metal using, carbon dioxide fixing. That is a profile. And that profile, as the researchers noted, is consistent with an alkaline hydrothermal vent setting. Consistent with, not proven by. The genomic reconstruction does not say Lost City, it does not name serpentinization or carbonate towers. It describes a set of metabolic preferences and environmental tolerances, and researchers point out that those preferences and tolerances fit the alkaline vent environment in several specific ways that are worth understanding. The hydrogen comes from the rock itself. In the serpentinization reaction, water penetrates ancient mantle rock and reacts with the iron and magnesium silicate minerals there, converting them to serpentine and releasing hydrogen as a byproduct. That hydrogen drifts upward through the vent system and out through the porous mineral walls of the towers. It is a constant, geologically reliable supply, driven not by volcanic activity that can shut off unpredictably, but by a slow, steady rock-water chemistry that at places like Lost City has been running for tens of thousands of years. An organism that evolved to run on hydrogen in such a setting would have a fuel source that was as reliable as the seafloor itself. The carbon dioxide came from the ancient ocean. Before the rise of photosynthesis, before life had done its long work of transforming the atmosphere, the early Earth's oceans held far more dissolved carbon dioxide than they do today, That carbon dioxide-rich seawater, slightly acidic, pressing against the alkaline hydrogen-rich fluid rising through the vent, would have created a gradient at every mineral wall, a proton gradient, a difference in acidity between one side of a thin surface and the other. And that gradient is the thing that makes the hair stand up on the back of your neck if you have spent any time thinking about cellular biology, because it is exactly the kind of gradient that every living cell on Earth uses to make energy. Let us sit with that for a moment. In 1978, a British biochemist named Peter Mitchell received the Nobel Prize in Chemistry for a discovery he had spent years fighting to have accepted. The discovery was chemiosmosis, and the resistance he encountered from the scientific establishment before it was finally recognized tells you something about how genuinely unexpected the idea was. Before Mitchell, the prevailing assumption was that cells made energy, specifically that they synthesized the molecule adenosine triphosphate, ATP, the universal cellular currency, through a series of direct chemical reactions, the kind where molecule A reacts with molecule B to produce product C and release energy. Clean, comprehensible, mechanical. What Mitchell showed was stranger. What cells actually do is use the energy released by metabolic reactions to pump protons, hydrogen ions, across a membrane from one side to the other. They build up a concentration gradient. They create a proton-rich environment on one side and a proton-depleted environment on the other. And then they allow those protons to flow back across the membrane through a remarkable protein machine called ATP synthase, and the flow of protons spinning that machine is what drives the synthesis of ATP. It is, in a very real sense, a turbine, not a chemical reaction in the ordinary sense, but a flow of particles through a machine, like water turning a wheel. And this is what every living cell on Earth does. Bacteria do it. Archaea do it. The mitochondria in your own cells do it. The chloroplasts in plant cells do it. The mechanism is so deeply conserved across all of life, so ancient, so foundational, that it appears to have been present at or very near the very beginning. It is not one metabolic strategy among many. It is the metabolic strategy, the one that everything else is built on top of, which is why the alkaline vent hypothesis has a particular resonance that is hard to dismiss, even if you are skeptical of it. At an alkaline vent like Lost City, a proton gradient forms automatically. The alkaline fluid rising through the vent has fewer protons than the acidic ocean pressing against it from the outside, across the thin mineral walls of the vent pores, walls that are, in some cases, only a few nanometers thick, comparable in thickness to a biological membrane. There is a natural proton gradient running continuously for free, driven by nothing but chemistry and geology. No cell required. No biological machinery needed to establish it. The hypothesis, as Michael Russell proposed it originally and as William Martin and Nick Lane have developed it with great care and considerable mathematical rigor, is that life did not invent chemiosmosis. Life inherited it. The proton gradient was already there, running in the mineral pores of an alkaline vent field on the early Earth, and the first protocells did not need to figure out how to generate a gradient. They only needed to learn how to harvest one that was already in place. The first trick was not creating the wheel. The first trick was learning to let the current do the turning. The porous mineral structure of the vent walls matters enormously in this argument. Those pores and channels in the carbonate and iron sulfide minerals are not simply passages for fluid. They are compartments. They are enclosures. They are spaces with an inside and an outside, with fluid composition that differs between the interior and the surrounding ocean, with reactive mineral surfaces on their walls that can catalyze chemistry. Iron and nickel sulfide minerals in particular have been shown in laboratory experiments to catalyze some of the same reactions that modern metabolic enzymes catalyze. They do biochemistry very slowly and imprecisely, using only rock. The hypothesis suggests that those mineral compartments were the first cells, or at least the first cell-like environments, and that the chemistry of life assembled itself inside them before biological membranes evolved to replace the mineral walls and allow cells to detach and move freely into the open ocean. This is the picture the hypothesis paints, and it is internally consistent and chemically grounded and connects the universal biology of chemiosmosis to a geological process in a way that is, at minimum, a genuinely serious scientific argument. It is the work of serious people, Russell, Martin, Lane, who have spent careers developing it with rigor and who have responded to criticisms and refined the model and published it in major scientific journals. And it remains, as of now, a leading hypothesis in a genuinely open field. Not the answer, a very serious candidate, because the warm little pond is also a serious candidate. Darwin wrote the phrase in a letter in 1871, almost casually, imagining a warm pool with the right chemicals stirred by sunlight and occasional lightning. He did not develop it into a formal hypothesis, but the image persisted, and researchers, including Bruce Damer and David Deamer, have done serious, careful, experimental work on what happens when the kinds of organic molecules found in meteorites are introduced into warm pools that go through wet-dry cycles, filling and evaporating, with each cycle concentrating the molecules, allowing them to react, and occasionally forming structures that begin to look like primitive membranes. The wet-dry cycle does real chemistry. The experiments are real. The alternative is real. And the land-based hydrothermal spring scenario, associated with Armand Mulkijanian and others, points to the specific mineral chemistry of geothermal pools on land as a better match for the ionic environment inside living cells. The relative concentrations of potassium and sodium and other ions that life, oddly, maintains at ratios that look more like some land-based geothermal fluids than like seawater. That argument is also based on real evidence and real chemistry. The question of where life began is open. This is not a failure of science. It is a fair reflection of how difficult the question is, and how old and how complete the erasure is of whatever happened in those first hundreds of millions of years when the chemistry of living matter was assembling itself in the dark or the light or somewhere between. The rocks that might have preserved that story have been subducted, overturned, metamorphosed, erased by the very geological activity that also built the planet. We are working backward from the biology of creatures alive today to reconstruct an event that happened perhaps 4 billion years ago in an environment that no longer exists, under conditions we can only partially reconstruct from geological inference and chemistry. What we have is a set of clues, and among those clues, the deepest and most universal one, the proton gradient, the turbine, the chemiosmosis that Mitchell described and that the Nobel Committee recognized in 1978, points somewhere that is geologically real and still accessible to study. And that somewhere is a field of white carbonate towers on the floor of the Atlantic Ocean, still venting hydrogen and methane, still running its quiet chemistry in the dark, still building its towers millimeter by millimeter through the slow reaction of ancient mantle rock with seawater. Lost City is not just a metaphor or a hypothesis, it is a place. It exists. It is perhaps 60 meters of white stone rising from the Atlantis Massif, still active, and researchers have visited it and taken samples and run experiments and argued about what it means. And what it means at the very least is that the kind of environment the alkaline vent hypothesis requires is not theoretical. It is demonstrably possible, and it is running today, which is where the argument opens out into something very large, because if life could begin in an alkaline hydrothermal vent field at the bottom of Earth's ocean, in the dark, fed only by the chemistry of rock and water, with no sunlight required, no photosynthesis, no surface, no atmosphere, then the question of whether life exists elsewhere in the solar system becomes a profoundly different question than it was before 1977. Before 1977, before Alvin descended to the Galápagos Rift and came back up with photographs of something impossible, the assumption was roughly that life needed light, and therefore a surface, and therefore a planet within a certain distance of a star. In the habitable zone, as it is called, the band of orbital distances where water can be liquid on a world's surface. The planets of the inner solar system, perhaps. Worlds like Earth, bathed in starlight, with liquid oceans exposed to the sky. But vent life does not need the sky, it does not need the star, it needs rock, and it needs water, and it needs the right chemistry at the interface between the two. And there are places in this solar system where those things are available, in the dark, under ice, in oceans that have never seen sunlight, that may never have needed to. The moon of Saturn, called Enceladus, is only about 500 kilometers across. Small enough that you could drive across it in a few hours if it had roads, which it does not. It is covered in ice. And it is, as far as we currently know, the most dramatic demonstration in the solar system of the kind of geological activity that could sustain vent life. At its south pole, the ice is fractured into long parallel cracks called tiger stripes, and from those cracks enormous plumes of water vapor and ice particles erupt into space. The moon is venting its own interior ocean into the darkness around Saturn continuously in geysers that reach hundreds of kilometers above the surface. NASA's Cassini spacecraft spent 13 years in the Saturn system, and in its final years it flew through those plumes, not past them, through them. It dipped into the spray erupting from the interior ocean of another world and tasted what was there. And what was there was remarkable. In data published by Waite and colleagues in the journal Science in 2017, Cassini's instruments detected molecular hydrogen in the plume material. Molecular hydrogen, H2, two hydrogen atoms bonded together, the same hydrogen that serpentinization produces, the same hydrogen that Lost City vents in steady streams from its carbonate towers. The detection of molecular hydrogen in the plume of Enceladus, the researchers noted, is most readily explained by ongoing hydrothermal activity at the rocky seafloor of Enceladus's ocean, water reacting with rock just as it does at Lost City, releasing hydrogen into the water above. The same year, and in subsequent work published by Postberg and colleagues in Nature in 2018, Cassini detected complex organic molecules in the plume material. Not simple carbon compounds, complex ones, with masses and structures suggesting they were fragments of larger organic material being carried up from the ocean below. Water, hydrogen, organics. The three things that, together in the right setting, chemosynthetic life depends on. This is not proof of life. It is emphatically not proof of life, and it would be wrong and careless to say otherwise. What it is a detection of chemistry. A detection of chemistry that, on Earth, is the chemistry that vent life uses, found now in the plumes erupting from a tiny, ice-covered moon in the outer solar system, from an ocean that has never been touched by sunlight, in the darkness beyond the orbit of Jupiter. And slightly further than that, in a different direction, a much larger world waits. Europa is one of Jupiter's 4 large moons, first observed by Galileo in 1610, and it has been known for some decades now to harbour a liquid ocean beneath its ice shell. But the scale of that ocean is still sometimes surprising when you encounter it clearly. Europa is estimated to hold roughly twice the volume of water contained in all of Earth's oceans combined. Twice. All the water in the Pacific, the Atlantic, the Indian, the Arctic, the Southern Ocean, all of it. And then the same amount again and more. All of it liquid, all of it in the dark, beneath a shell of ice that may be several kilometers thick, in an ocean that extends perhaps 100 kilometers deep. If that ocean has a rocky seafloor, and the current understanding of Europa's structure suggests that it does, and if the tidal forces that Jupiter exerts on Europa generate enough heat to drive any hydrothermal activity at that seafloor, then you have, in principle, a setting: rock, water, heat, and chemistry. The ingredients. Not the conclusion, the ingredients. NASA's Europa Clipper spacecraft launched in October 2024 with the goal of investigating whether Europa's ocean could be habitable. It is not a life detection mission precisely. It cannot land, it cannot sample the ocean directly. What it can do is fly close to Europa dozens of times, measuring the composition of whatever material is erupting or sputtering from its surface, mapping the ice shell, characterizing the magnetic environment, looking for signs of chemical exchange between the ocean and the ice above. It will tell us, with far greater precision than we currently have, whether Europa's ocean is the kind of place where life might be possible. It will not answer the question, it will sharpen it. And the question it is sharpening is the question that Alvin answered for us in February 1977 in the most unexpected way imaginable. The question is, does life require the sun? The answer we discovered at the Galápagos Rift in the dark, 2.5 kilometers down, in water that had not been touched by light since it fell as rain perhaps thousands of years before, is no. Life does not require the sun. Life requires chemistry, and chemistry happens wherever rock and water meet in the presence of energy, which at the bottom of a sea, whether Earth's sea or Europa's or the hidden ocean of Enceladus, is exactly the kind of energy that geology provides for free. But there is a quiet note beneath all of this that is worth acknowledging before the wider wonder takes over entirely. The vents themselves, the lost cities and the black smokers, and the alkaline towers venting hydrogen into the ancient dark, are not simply philosophical subjects. They are physical places, they are ecosystems. They support communities of life that exist nowhere else on Earth, that evolved in conditions so specific and so isolated that the animals living there are not found anywhere else in the world. The scaly-foot snail with its iron armor and its enormous heart, the Pompeii worm with its bacterial fleece and its extraordinary gradient, the tube worms with their billion per gram bacterial farms and their impossible chemistry, the yeti crab waving its gardens over the flow, the blind shrimp turning its back-mounted sense organ toward the faint glow of heat. These communities, these impossible and irreplaceable ecosystems, are under threat from the same geological wealth that makes them interesting to scientists. The mineral chimneys that vent life builds around and within are rich in copper, zinc, gold, and rare metals. The polymetallic sulfides that are being surveyed and in some cases targeted for deep-sea mining. The scaly-foot snail, Chrysomallon squamiferum, was listed on the IUCN Red List as Endangered in 2019, the first deep-sea vent species to receive that designation. Not because it has been hunted or fished or poisoned, but because its entire known range consists of a tiny number of vent fields, and those vent fields sit above mineral deposits that mining operations have proposed to extract. The places where the most extraordinary life on Earth persists are also the places where economically valuable minerals are most concentrated. That is not a coincidence. It is the same chemistry, seen from two different directions. And those places may also be the places most likely to inform the oldest question we can ask about ourselves: where we came from, what the first living thing was, how chemistry became biology in some corner of an ancient sea that no longer exists and left no direct record of itself, only the faint genomic shadows of its descendants in every cell of every creature alive today. The 355 gene families that Weiss and colleagues reconstructed. The proton gradient running across a mineral wall in a vent field that has been venting for 30,000 years. The strange blood of a tube worm that carries a poison and an antidote at once, bound to the same molecule in a physiology that would have seemed fantastical before 1977. All of it pointing, gently and incompletely, toward the same answer to the same question. It is worth pausing here on the strangeness of that convergence because it is easy to let it pass by when you are listing facts and dates and chemical names. The thing to notice is not any one of these pieces separately. The thing to notice is the thread running through all of them. The energy mechanism in your cells, the proton gradient, the spinning turbine of ATP synthase, resembles a process that happens for free in a field of white carbonate towers at the bottom of the Atlantic Ocean. The organism from which all life on Earth appears to descend may have lived in exactly that kind of environment. The animals living there today are the closest things we have to a working model of how life can thrive in total darkness on chemistry alone. And the moons of the outer solar system show us that rock, water, and the right chemistry are not unique to Earth. The deep sea is not the edge of the world, it is, perhaps, the center of it. The place where the most fundamental things about living matter are most plainly visible, unobscured by the noise and complexity of the sunlit world above. And the truth is that we are still learning to read it. Alvin descended to the Galápagos Rift in February 1977 with no biologist aboard and came back up with photographs of something no one had a framework to explain. 40-some years of research later, we have Riftia's biochemistry and Lost City's geology and Luca's reconstructed genome and Cassini's passage through the plumes of Enceladus, and we still have not reached the bottom of the question. We have only found better tools for asking it. More precise measurements, longer dives, a spacecraft now traveling through the outer solar system toward a moon with twice Earth's worth of ocean beneath its ice. The question is still open. It is open in a way that is, if you let it, restful. The not knowing is not a failure. It is the shape of a real question, the kind that only gets asked when you have found something genuinely large enough to matter. Somewhere beneath the ice of Enceladus, if the hypothesis is right, if the chemistry detected by Cassini means what the researchers who have spent years analyzing it believe it most likely means, there may be a rocky seafloor where hot alkaline fluid rises through serpentine channels in ancient mantle rock, and where mineral pores and chambers line the vent walls, and where the same chemistry that built the first chemistry here is running quietly in the dark. Whether anything has yet learned to harvest it, whether biology has yet arrived at that chemistry as a resource rather than simply a process, we do not know and cannot yet know and may not know for years or decades or longer. But the question is real, and it is the same question that the worm with no mouth raises, and that the snail in its iron armor raises, and that the Pompeii worm, raising each end of its body into a different temperature, raises, and that the blind shrimp, turning its back to the heat, raises. The question of what life is fundamentally underneath all its strange and particular elaborations, underneath the hemoglobin and the greigite and the trophosome and the bacterial fleece. What it runs on, where it started, whether it is here alone or also elsewhere, in other dark oceans, under other ice shells, around other stars. That question was asked by the geology of this planet before any living thing was present to ask it. And it is still being asked in the white towers of Lost City, rising 60 meters from the floor of the Atlantic in the dark, patient as stone, venting their hydrogen and their methane into the cold water above, as they have been doing for 30,000 years or more. Let that sit for a moment. 30,000 years of continuous chemistry. The first cave paintings in Europe are perhaps 20,000 years old. Lost City was already old when human beings first put ochre to stone and drew the animals they hunted in firelight. The question it is asking has been running longer than we have, and it is asking it still. The deep sea floor right now, as you listen to this, is not still. It is alive in the way that the most permanent things are alive, slowly, continuously, without urgency, without audience. The Riftia colonies, swaying in the warm vent currents. the scaly-foot snail moving across the rock with its iron scales catching whatever faint mineral light the hydrothermal fluid produces. The Pompeii worms in their tubes, their tails in the heat and their heads in the cold, eating the bacterial fleece that grows on their backs. The yeti crabs waving their arms. The blind shrimp turn toward the warmth they cannot see but have learned to feel. All of this happening in complete darkness, kilometers below the surface of the sea, in water that photons have never reached, sustained by chemistry that the planet provides and always has provided, since long before there was any living thing to use it. And the thought to carry with you, if you carry anything from this into sleep, is simply this. The strangeness is not the exception. The strangeness is the thing being pointed at. Every one of those impossible animals is a working answer to a question we barely knew to ask before 1977. They are proof that life finds a way, not as a slogan, but as a biochemical and geological fact demonstrated in the sulfide-binding hemoglobin of a worm's blood and in the iron sulfide sclerites of a snail's foot, and in the proton gradient running across a mineral wall in the North Atlantic in the dark. And if life finds a way here, in the strangest corner of one world, then the question of whether it finds a way elsewhere becomes not a fantasy, but a reasonable scientific possibility. A question worth asking with real instruments and real spacecraft and real decades of patient, careful work, which is what we are doing. Europa Clipper traveling now through the outer solar system toward a moon we have never touched. Cassini's data still being analyzed, still yielding surprises from the archive of its 13 years in the Saturn system. And on Earth, in laboratories and in submersibles, and in the cold waters near hydrothermal vent fields, researchers continuing to ask what the chemistry means and what the biology says about the chemistry, and what the genomics says about the biology, and what all of it taken together says about the morning of this world. You do not need to hold any of it tightly tonight. The questions are older than you are and will outlast the night. The towers of Lost City are standing, the worms are in their tubes, the snail is moving across the dark rock in its iron coat, the plumes of Enceladus are still erupting into space, carrying their hydrogen and their organics out into the cold between the planets, patient and indifferent and continuous. Let the deep sea carry it now. Like the old Stoics, let the day go. Tomorrow can be tomorrow's.